Welcome to Anatomy series. This course deals with general histology. Today we are going to discuss about the epithelial tissue. What is a tissue? A tissue is made up of group of cells performing the same function, and the different types of tissue are epithelial tissue, connective tissue, muscular tissue and nervous tissue. These are the questions asked in exam. Various types of tissue. This chapter deals with epithelial tissue. The epithelial tissue, it covers the outer aspect of the body and it lines tubes and tracts. So in this diagram we can see it is lining the inner aspect here in this tube GIT, and this is the covering the outer aspect, that is in skin. Functions. The functions of epithelial tissue are protection. The stratified squamous epithelium, which is on the skin, it offers mechanical protection. Next function is secretory function. The glands, which are derivatives of epithelium, they secrete various substances which are useful. Absorptive function. The epithelia of small intestine and proximal convoluted tubules of kidney, we know that they are modified and they have a special absorptive function. Excretory function. The epithelium of certain tubules, like again collecting ducts of kidney, they function as excretory organs. Sensory function. The rod and cone of retina, they have got specialized sensory cells. So these are the functions of epithelial tissues. The features of epithelial tissue. The epithelium is either made up of a single layer or many layers. The cells are joined of, here if you can see, they are adherent to each other by means of junctional complex. These cells, they lie on basement membrane. Basement membrane is of two parts, basal lamina and the reticular lamina. Basal lamina is a part of the epithelium, while the reticular lamina is part of the basement membrane. Epithelium is derived from all the three germ layers. Skin is derived from ectoderm. Respiratory and digestive system, they are derived from endoderm, whereas the cardiovascular system is derived from mesoderm. Let us now discuss about the junctional complexes. We said that the epithelial cells which are close to each other, they are adherent by junctions, and these are known as the junctional complexes, or they are known as intercellular junction. Here there are certain molecules known as cell adhesion molecules, or CAM. They are found in between the plasma membranes of the adjacent cells. These molecules are formed by glycoprotein and proteoglycan. Also, in addition to this binding effect of the CAM or the cell adhesion molecules, calcium ions also play an important role. The four junctional complexes are: first, zona occludens, which means tight junction. This junction is usually near, or it is usually located near the apical part of the cell. This one is a tight junction. Zonula adherens. This is again a junction which is present just below the zonula occludens. In this diagram you can see zonula occludens is tight, there is no gap in between. But here in zonula adherens there is a slight gap, that is 20 nm wide. Zonula adherens, it is adherent. It is said that here there are certain filaments which help in exchange. Then there is macula adherens or desmosomes. They are scattered over the again lateral surface of the epithelial cells. Now, if you can see here, this is a desmosome. The plasma membranes here, they are separated by a gap of 30 nm, whereas we saw just now that gap was 20 nm only. This is 30 nm, so that is, this gap is slightly more, and this is bridged by transmembrane proteins. There is something known as hemidesmosomes, that is half desmosome, and they are found here on the basal surface. So, macula adherens means desmosome. Desmosome, full desmosome, if you can see, they are near the adjacent cells, and hemidesmosome is half, which lies at the basal surface. Gap junctions. The word itself says gap. So, here the two cells or the adjacent plasma membranes of the two epithelial cells are opposed, and there is a presence of gap. If you can see here, there is a gap in between them. In this diagram, if you see, there is zonula occludens in the topmost part, apical part, then there is zonula adherens, then there is macula adherens, and then inferiorly if you go, there is gap junction. Gap junction, in this there is easy exchange of messengers and chemical messengers and the nutrients. This is in a tabular form to explain it in one slide itself. The tight junction, zonula occludens, luminal end, zonula adherens, lateral part of the plasma membrane, just below the zonula occludens, desmosome, hemidesmosome, basal plasma membrane, and gap junctions. Okay. Now we come to the next part, that is the epithelial cells modifications. The surface of the epithelial cells, they show certain modifications depending upon the action. So, it is glycocalyx, microvilli, stereocilia and cilia. Stereocilia and cilia. Let us see one by one. Glycocalyx. Glycocalyx क्या है? It is a surface coat or over the absorptive epithelium. Wherever there is absorption is required, there we find this glycocalyx. It acts as receptor site for hormone and enzyme. So, where it can be? It is seen mostly in the small intestine. Then there is microvilli. Microvilli is brush border. Microvilli, these are minute finger-like projections of the plasma membrane. They increase the surface area for absorption. So, this is also seen in intestine. It is said that they also participate in digestion of carbohydrates. So, this is the microvilli. This is finger-like projection, minute finger-like projection, and if you see here, glycocalyx is surface coat over it. So, if I am taking a microvilli cut section, over that I can see glycocalyx. Okay. Then there is stereocilia and cilia. Stereo is, in Latin it means solid. So, stereocilia, they are long, thick microvilli. They increase the surface area for absorption, but they are non-motile. So, stereocilia are non-motile apical cell modification. They are found in vas deferens and the epididymis, also in the sensory cells of the inner ear. They help to increase the surface area for absorption. Whereas cilia, these are long hair-like projections of plasma membrane. They beat in one direction. So, their main function is pushing the particles towards one direction. For example, in respiratory tract, they beat towards the pharynx, and in uterine tube, they beat towards the uterus. So, this is a cilia where they are actually beating the function Their function is to beating towards one direction. Whereas here, if you can see, stereocilia non-motile. Cilia okay, this is the difference between a microvilli and cilia. Microvilli, we have seen this finger-like projection, minute finger-like projection, where here we have seen long hair-like projection. Absorption, mostly the function is absorption. Here transport is the main function. Classification of epithelium, another important part in epithelium. The epithelium can be single layer, then it is known as simple epithelium. This simple epithelium, depending upon the type of cell, they are again classified as squamous epithelium, which is flattened. If the cells are cuboidal in shape, then it is cuboidal epithelium. If the cells are columnar, it is known as columnar epithelium. Stratified epithelium is next classification, which means multiple layer. More than one layer, it is stratified epithelium. So stratified, if the cells are squamous, it is known as stratified squamous or stratified cuboidal epithelium. Transitional epithelium is a stratified epithelium, but it is classified separately. We will see this again in detail about the transitional epithelium. Okay, this is to show you our squamous epithelium. It is flattened. The nucleus is flattened. Mainly we see the nucleus in the slide. If the nucleus is flattened, means the cell is flattened. See. Here if you see, the nucleus is rounded and in the center. It is usually said that the length and breadth of the epithelial cell is same, and then it is known as cuboidal epithelium. Here breadth is more, length is small, then it is a squamous epithelium. The length of the cell is more, breadth is small, and it is columnar shaped, so it is a columnar epithelium. See the nucleus. Nucleus is also looking columnar. So this is a columnar epithelium. This is columnar epithelium with cilia. Stratified epithelium, as we said. Stratified squamous, and it can be keratinized if the keratin is present over it, and it stratified squamous, non-keratinized. So multiple layer with keratin, multiple layer without keratin. Stratified cuboidal. The cells are cuboidal in shape, multiple layer and cuboidal in shape. What will stratified columnar mean? Columnar cells but multiple layer. So stratified columnar. Transitional. Transitional epithelium, if you can see, again it is a multi-layer epithelium. Now if you see in detail, this transitional epithelium is a stratified epithelium, but it is classified separately, because if you can see, the cells here are binucleate and they are typical umbrella shaped cells in the topmost layer. And the other layer, it is pear shaped. These cells, they have a special quality that they are impermeable to the urine present in the lumen. ब्लैडर में यहाँ पे अगर यूरिन है तो ये एब्जॉर्ब नहीं होता है. Okay. Also it has got a quality that it can stretch and it can form, umbrella shaped, and when it is relaxed it comes back to form the pear shaped. So transitional epithelium is a multi-layer epithelium or a stratified epithelium with having specialized cells which are umbrella shaped and pear shaped. This epithelium is found only in the urinary bladder and the ureter. Pseudo-stratified epithelium. Pseudo is false and stratified is multi-layer. Means the epithelium is single layer but it falsely appears to be multi-layer. This happens when the nuclei are at different level. If you can see here, there is a single cell, but the nucleus is lying at different levels and it appears to be stratified or multilayer. But so this is known as pseudo-stratified epithelium. This pseudo-stratified epithelium, it can be ciliated and then it is known as pseudo-stratified ciliated epithelium. ये कॉलमनर सेल्स है, elongated nucleus है, इसके लिए pseudo-stratified columnar ciliated epithelium found in nasal cavity and trachea. Pseudo-stratified columnar epithelium with stereocilia, epididymis and vas difference. Stereocilia non-motile. So this is stratified squamous non-keratinized and this is pseudo-stratified ciliated, just to show you how the nucleus is appearing at different levels. Simple columnar ciliated. This is how it will look in the histology. This is seen on high power. Elongated nucleus and cilia hai. Transitional epithelium, umbrella shaped epithelium. If you can see here, stratified hai, but a peculiar epithelium seen. Urothelium also it is called as. Applied anatomy. There is something known as metaplasia and dysplasia. Dysplasia term is used to describe the presence of abnormal cells. Dysplasia is not carcinoma, but it can convert itself into carcinoma. So abnormal cells दिखते हैं. Whereas metaplasia is one cell, it has to form a particular cell, but this cell, it forms itself or it converts and forms into some other cell, and this can be reversible. So this is less likely to lead to cancer. Or if you can see here how it is given, conversion of a cell into another form of cell upon injury or insult. Here it is given dysplasia is development of abnormal type of cells within the tissue. Means metaplasia ke liye there is a external stimulus, but here there in dysplasia there is a internal stimulus. This is metaplasia is reversible, dysplasia is irreversible. Metaplasia do not lead to formation of carcinoma or cancer, but dysplasia can form cancer. तो अगर यहाँ पे देखेंगे ये नॉर्मल सेल है, अगर ये सेल साइज इंक्रीज हुआ तो इसको हाइपरट्रॉफी कहते हैं. अगर नंबर ऑफ सेल्स इंक्रीज हो गया तो हाइपरप्लेजिया कहते हैं. मेटाप्लेजिया एज बी सेड, external stimulus, dysplasia and hyperplasia. This is hypertrophy and hyperplasia. This is hyperplasia only, and this is atrophy. Cell size is also decreasing, not the number. Thank you.